Hello everyone! After our discussion of inheritance and looking at simple Mendelian traits, we're now going to jump ahead and we're actually going to start talking about Chapter 7, which is all about the influence of the environment on our traits. So this is our Genes and Environment lecture. So we're going to talk about a couple of different things. So after learning about those simple Mendelian traits, we're going to talk about all of the other traits we have. So we're going to talk about polygenic traits versus multifactorial traits. We'll take a look at different examples of those, and then we'll also look at some tools that we use to analyze these multifactorial traits. So let's first off talk about this idea, and I kind of mentioned it in Chapter 4, that there are actually very few genes that we have that are fit into this nice, neat pattern of being, again, single gene traits, also called Mendelian traits. In reality, most genes don't act alone. There are other genes that influence a trait collectively, and environmental factors can also play a big role in our traits. So we're going to use a little bit of terminology here to describe three different types of traits, one of which we've already covered and two of which we're going to cover in this lecture. So again, Mendelian traits are any trait that's caused by single gene. We say single gene, single action. So those were the things that we were looking at, the traits, the pea color traits, or the traits in pea plants, for example. Today we're going to talk about polygenic traits and multifactorial traits. Polygenic traits are caused by multiple genes acting together, and multifactorial traits are traits that are interactions between genes and our environment. Alright, so let's talk about polygenic traits first. So this is a trait that's due to multiple genes working together to produce some type of a trait. And what's important to know about polygenic traits is the effect of the genes is additive. So what that means is the more genes you have for, let's say, skin color and melanin production, you've got 4 different genes that are responsible for the pigment in your skin. The more of those genes that you have that produce melanin, that produce that dark color, the darker your skin is going to be. So again, it's an additive effect. That's the idea. Genes can have either major or minor impacts. So again, in the case of skin color, we're looking at that additive effect. The more melanin you have versus the whitish color, which is called eumelanin, the darker your skin can be. And what we see is a continuum of phenotypes. So unlike in the most Mendelian traits that we were talking about, like the diseases, right? You either had cystic fibrosis or you didn't, or you could have been a carrier, I guess. That was kind of an option, but there's no phenotype there, right? Going back to the pea plants, you were tall or short. You had purple flowers or you had white flowers. There was nothing in between. Polygenic traits produce a continuum of phenotypes. So there are variations in between, if you will. And even though the individual genes of a trait follow Mendel's laws, if you put them all together, you don't produce those Mendelian ratios. So I talked a little bit about ratios kind of at the very end of that first lecture, but what's important to know is you don't always get if you're looking at, a heterozygote, and we know if we cross 2 heterozygotes, then, 75% of the offspring will have the dominant phenotype. It doesn't work the same way with these types of traits. So let's take a look at an example here, and we're going to use eye color. And this is a slightly outdated model for eye color, and I'll tell you that right off the bat. But we used to use this old 2-gene model for inheritance. And essentially, the first gene, you could have one of 2 alleles, either a brown allele or a blue allele. And the second gene, you either had a green allele or a blue allele. So brown was dominant to blue, green was dominant to blue, and brown is dominant to everything, both green and blue. So if you were to take a look at sort of different combinations that could be produced, what you see is that if you had a single brown allele, regardless of what you had at the other gene, you're going to have brown eyes. The only way you could get green eyes then was if you got 2 blue alleles in the first gene, and because green is dominant to blue, you would have had green eyes. And the only way that you could get blue eyes if you had all recessive alleles, so blue across the board. So again, it's an outdated model, but it kind of shows you this idea of what could happen if you have two genes influencing a particular trait. We think there might be at least 4 genes for eye color, maybe 2 for what we call the lipochromes and 2 for the melanins. And the reason is this. Not all blue-eyed people have the same shade of blue, right? There are a whole host of shades of blue eyes. Same thing with brown eyes, same thing with green eyes. So to say that there's just these 2 genes is a really outdated model. We could find more genes. There are 60 eye color genes in mice. So that tells us that there potentially could be a lot more genes at play here. We just haven't identified them yet. So let's move on to the next category of traits, and these are the multifactorial traits, which we're going to spend most of this lecture talking about. And these are traits that result from an interaction of genes and the environment. No Mendelian ratios here. We can't use that at all. We can predict phenotypes using a couple of different tools. We can predict phenotypes using something called risk analysis, which I'll talk about, family history, the occurrence of the trait in the population. So there's certain things that we can actually use some tools for predictive measurements here. But things like fingerprints, your height, your skin color, your risk of heart disease, your risk of diabetes and cancer, and all of these traits specifically related to our health are all examples of multifactorial traits. And again, it's important to note that we have what's called continuous variation. And again, we use this for both those polygenic traits and these multifactorial traits. And basically what it says is if you were to take individuals in a population and you were to plot their phenotypes, okay, so whatever trait we're looking at, let's say it's height. Okay, so we're doing height. Height is a good example of a multifactorial trait. What you'd find is you have some people that are on the very small end of this scale, so some shorter people. Most people are going to fall into what's known as our average, and then there are a few people on the end here who are going to be taller, right? So this is our continuous variation. There are phenotypes in between here, right? That's the idea. So again, height, skin color, body weight, hair color, cholesterol levels, you name it, all of these would fit this pattern, if you will, where we see continuous variation in a population. So I'm going to kind of breeze through a couple of these, but your fingerprint pattern is both a multifactorial and a polygenic trait. The number of ridges you have is determined by your genetics, but the pattern that you have is actually determined by when you're in the womb, how much and what you are touching between 6 and 13 weeks of development. So it's kind of very interesting. So your womb environment plays a direct role in determining what pattern you have. And here are some example patterns that you can have. So again, if you were to look at, this is a study that basically a professor did on his classroom, and he looked at the number of students he had, and he looked at total ridge counts, so the number of little lines that we see here. And once again, you can kind of see that continuous variation, right? He had a few students who were sort of on the lower end. The majority of students were here in the middle, and a couple of students had a high pattern. So once again, continuous variation that we see in these traits. We can do the same thing for height. Again, these are 2 different pictures, basically, one taken with military cadets in 1914 and another done with college students about 75, 80 years later. Continuous variation, that's what we see. A couple different things play into height. So what we see is that in the 2 groups, if you look carefully at that picture, which I know I breezed through, but you can go back and pause that if you'd like, what you'll notice is that the overall average increased, as well as the tallest individual, the height of the tallest individual, and that's due largely, in fact, to overall health and better diet in between the 2 groups. So we know that there are dozens of genes that affect height, and again, also your nutrition plays a role in that. So especially as a child, as a growing child, if you're not receiving the proper nutrition that you need, you're not going to be able to reach what we call your height potential, and that's based on your genetics. Body weight is another example of a trait that is multifactorial and, again, polygenic. There are lots of traits involved. And we know that in the United States in particular, we're in the midst of basically a health crisis, in the sense that we have an awful lot of adults who are overweight or obese. And if you look at this measure of BMI, if you've never looked at one of these charts before, so your body mass index is sort of an overall a number that can tell us, like in an instant, kind of a snapshot of what your weight health is, if you will. And again, it's not perfect and it doesn't work for everyone, but in general, you find your height, you find your weight, and you find your BMI. And depending on where it falls, you're healthy, you're overweight, or you're obese. That's the idea. So we know that using some twin studies, which we're going to talk about a little bit later on, we know that there's a heritability here of about 75% for obesity. So what that means is there are definitely genes at play here that are leading to some individuals to be obese, or it's harder for them to basically take that weight off. But we also know that your environment plays a direct role. Your lifestyle, your diet, your exercise, all of those things can play a role. Genes influence things like hunger and metabolism. So some people really do have slower metabolisms and some people have quicker metabolisms. For some people it's hard to lose weight and for some other people it's easy to lose weight. But you can see here how obesity trends have changed over the past 30 years, and it's got nothing to do with genes, right? Our genes haven't changed over the past 20 to 30 years. What's changed is our diet and our lifestyle. So look at the obesity rates back in 1990 compared with 2000, compared with 2010. And again, this figure itself is outdated because it's 2019, and there are a lot more states in the red now than there used to be. So it's not our genes that have changed, it's our lifestyle that has changed, and that is what's leading directly to this obesity crisis. Other things, heart health, cardiovascular disease is another example of a polygenic trait and multifactorial trait. So again, depending on your diet and your exercise and your lifestyle choices like smoking and drinking and those types of things, all of those play into your cardiovascular health. Okay, let's do a quick comprehension check. Which of the following is not a continuously varying trait? Would it be your height, your cholesterol level, your weight, your ABO blood type, or your IQ? This one might be a little tricky. The answer is your ABO blood type, and I know that we didn't specifically talk about this trait, but if you think about it, there's no continuous variation here. You either have type A, type B, type O, or type EB blood, right? There's nothing in between, right? Whereas for all of the other traits, we have a variety of phenotypes that we can see here. All right, so here's the challenge. How do we know how much of a trait is due to an influence of the environment? And how do we know how much of a trait is due to our genes? Well, we have sort of this kind of generalized graph here, and if you look at this, this is actually causes of morbidity and mortality, so these are things that could cause you to die, and this is the relative contribution of how much of these factors come from genetics and how much of these are environmental. So things like cystic fibrosis and hemophilia and certain types of breast cancer and Alzheimer, all of these have a higher genetic component to them and a lower environmental component. But then when you look at things like smoking-induced lung cancer and motor vehicle accidents and gunshot wounds, very little of the environment here, or very little bit of genetics is involved. This is all your environment. So we need to have a way that we can actually tease these things out. And a couple of just vocabulary terms here. We talk about something called your empiric risk, and it measures the likelihood that a trait will recur based on incidence. So what is incidence? Incidence is the rate at which a certain event occurs. So how likely it is that you're going to die in a motor vehicle crash. That's your empiric risk of dying in a motor vehicle crash. What's your chance of dying from heart disease or something like that? And then we talk about prevalence being the proportion or number of individuals who have the trait at a specific time. How many people in our population have diabetes, type 2 diabetes? That's the prevalence of type 2 diabetes. So heritability is what we're after, and heritability estimates the proportion of the phenotypic variation in a population due to genetic difference. Okay, so what does that mean? So what we're looking at is we want to figure out how much of that trait is due to your genetics and how much of that trait is due to the environment that you are a part of. So we take a look at these multifactorial polygenic traits, and what we want to figure out is this part, the genetic variance. We want to look at the additive effects of alleles we want to look at dominant alleles. Don't worry about this epistasis. We're not going to discuss that. But this is what we're after in heritability. We know environmental variance happens. We know people who smoke and don't smoke. There's health differences between them. But what we don't know are these genetic variances. So we have calculated heritability for a variety of different traits. And what's interesting is the higher the heritability, the more genes are responsible, and the lower the heritability, the more environment is responsible. So clubfoot, which is a condition that you're born with, your height, your blood pressure, your verbal aptitude, your mathematical aptitude, interestingly enough, is not very heritable, so that's mostly your environment and not your genes. Your total fingerprint ridge count, we talked about that, that's highly heritable. So all of these things are what we're after with these, trying to pinpoint these multifactorial traits. So how do you figure this out? How can we statistically estimate heritability? One way we can do this is compare different populations of people. People who share the trait to the number of people who we think should share the trait. And the expected proportion is derived by knowing the blood relationships of the individual. So all of this goes into twin studies. So twin studies are great because we can take different groups of individuals, adopted individuals who share the exact same environment but have no genetic similarities, dizygotic and monozygotic twins. These are kind of the two groups that we study a lot. Dizygotic twins share an environment but only half of their genes. Monozygotic twins are identical twins, so they share both their environment and their genes. And then sometimes we can even get instances of twins raised apart where they share a genotype but they don't share an environment. So we use all of this information and we calculate something called concordance. So follow me for a minute here. Concordance measures the frequency or expression of a trait in both members of monozygotic and dizygotic twins. So if a trait is largely determined by genes, then we're going to see a higher concordance for monozygotic than dizygotic twins. Okay, so the idea is, once again, if identical twins share the trait more often than non-identical twins, then we know that there's a larger portion of our genes at play. All right. So here's some concordance values. Okay, remember, when we see a higher value here for identical twins and a lower value here, we know there's genetics at play. So acne, it was identical, right? So there's not a lot of genetics behind acne. Alzheimer's disease, definitely a difference here. There's genetics. Anorexia, genetics. Autism, look at autism. 90% concordance with identical twins. That means if one twin had the trait, there's essentially a 90% chance that the other twin was going to have autism as well. Whereas in fraternal twins, that goes down to 4.5%. So that tells us there's a huge genetic component to autism here. Bipolar disorder, cleft lip, hypertension, schizophrenia. So again, all of these things help us to determine that there's a large genetic component with these traits. I'm going to kind of skip over these last few slides, these genome-wide association studies. I'm not super interested if you know these. If you're interested, you can certainly go ahead and look at these, but again, I'm going to kind of skip over them. They're interesting. We're looking at something called single nucleotide polymorphisms, which I'm going to talk about later. And again, what it helps us to understand is it helps us to understand more about different types of traits, looking at large populations and not just twin studies, for example. But we're going to pass over that. Okay, so here are your important terms and important concepts to understand for Chapter 7.